Hello everyone, we are Team 5 and we are going to be talking about our article that's entitled Ribosome Biogenesis and Cancer.
We are Team 5 and the participants are Miguel Adame, Abril Carrillo, Oscar Sandoval, Mario Campo, Azul Vázquez and our producer Francisco Santos.
Okay, then let's start with our introduction. The history of the relationship between ribosome biogenesis and cancer begins long before the discovery of either ribosomes or the functions of the nucleolus in ribosome biogenesis. It was observed that cells of malignant tumors were characterized by particularly larger nucleoli than normal cells. The macronucleolus was considered unquestionably diagnostic for malignancy, and for a long period of time, the nucleolar hypertrophy was considered to be a cytological parameter useful for the diagnosis of malignancy. But in the 20th century, a series of studies showed that nucleolar hypertrophy, which frequently characterizes cancer cells, cannot be considered a reliable parameter of malignancy, and it is also observed in normal proliferating cells. A great contribution to this field was made not only by the introduction of new molecular techniques for the molecular investigation of cell functions, but also by the development of a histochemical technique for the visualization in situ histological routine preparations of the nucleolar structures engaged in the ribosomal RNA transcription. In very recent years, the interest in the relationship between ribosome biogenesis and cancer increased further after the finding that the rate of ribosome biogenesis controls the expression level of the tumor suppressor gene p53 and that an upregulated ribosome biogenesis is associated with an increased cancer risk.
Okay, now we are going to continue and talk about biogenesis. The ribosome biogenesis is the result of a series of coordinated steps. The transcription of the ribosomal DNA is going to occur in the nucleolus. It is going to require the assembly of a multiprotein complex, which includes the RNA polymerase or Pol-1 and the basal transcription initiation factors or just basal factors. In mammalian cells, there are going to be 3 basal factors, which are here. These 3. The first one, RRN3 or the ribosomal DNA transcriptional factor, SL1 or selectivity factor, and UBF or upstream binding factor. These 3 are necessary for the ribosome gene transcription. Transcription is going to be elongated by Pol-1, and the transcription is going to end by a release factor. The release factor is this one, PTRF. Okay, now we are going to talk about this molecular process. But before that, I'd like to explain the image. In the area that it's colored by a kind of aqua color, I put a letter N that is going to represent this area as the nucleolus. In this color that it's like white, I put a letter N and a P that is going to represent this area as the nucleoplasm. And where there's a light blue color, I put a letter C that is going to represent this area as the cytoplasm. The transcription is going to produce a 47S ribosomal RNA precursor. This one is going to be processed to generate 3 ribosomal RNA, which are 18S, 5.8S, and 28S. These are from the nucleolus. But there is a 4th ribosomal RNA, which is here, 5S. But this one is going to be produced in the nucleoplasm, and it's going to be produced by an RNA polymerase 3 or Pol-3. and after it's produced, it's going to be imported to the nucleolus. These ribosomal RNAs are then going to be assembled with ribosomal proteins. There are two, as we have in the image: orange of small units and green of large units. So when we form the small unit 40S, we are going to use the small unit ribosomal proteins, which are also called RPS, and the 18S. To form the large unit 60S, we are going to need the large subunit ribosomal proteins that I said are in green, and they're also called RPL, and the ribosomal RNA that are 5.8S, 28S, and the one that was transcribed from the nucleoplasm, that is 5S. Okay? The large and small subunit migrate to the cytoplasm, where they're going to finally make an 80S ribosome particle. And last, I like to say that in a cell, the rate of a ribosome production is going to be strictly dependent on pol I activity that I mentioned on the first slide, and on this one, that is the causes why the transcription is going to be elongated. Okay? Even though a pol III produces or transcribes an 5S ribosomal RNA, it doesn't depend on this one. It depends on pol I.
By now, you guys already seen the biogenesis of the ribosome, and I have some images right here so you guys can understand better. These images are taken with the electron microscope, and they tell us about the components of a cell. And here it was taken from a TG cell. A TG cell is a cancer cell of the fallopian tube, and this one right here, you can see it. This has several stuff. It has a pound sign right here, and this is the fibrillar center, and which is surrounded with a ring, and this ring is the fibrillar component, and all of this is merged into the cell. And well, all of this black stuff around it is called the granular component. And when we stain this cell with silver nitrate, we can get rid of this stuff around the cell, and we only have the center, the fibrillar center, and the fibrillar component. This image was taken with a light microscope, and as you can see, it was stained with silver nitrate as the other one, but here we have some pigments, and those dark dots are the nucleus organelle center proteins. And this is also called the NOR, and this NOR are the place where the ribosomal RNA is contained. In this first image, we can see the synthesis of the ribosomal RNA, and it is done in these green dots. The second picture reveals the unspecific fixed conjugated mononuclear antibody, and the third picture helps us to merge the the two images, so it looks like that. But to see better the structures, we have to split them. In this very last picture, we have real-time synthesis of ribosomal RNA, and the first one and the second one, we have almost the same amount. But when we get to the third one, we can see that it's lower than the other two. How do we know that? Because of this black pigment, which is the NOR, or the place where the RNA is contained.
Ribosome biogenesis and cell cycle progression. When a cell is stimulated to split, protein synthesis increases greatly so as to duplicate the cell structural and functional complements. The increased demand of protein synthesis is met by the upregulation of the ribosome biogenesis rate. Therefore, cell proliferation should be closely coordinated by the biosynthetic mechanisms leading to the formation of ribosomes. So at this point, together, all of these data indicate that the factors responsible for the stimulation of the cell proliferation also activate ribosome biogenesis. On the other hand, there is evidence that the ribosome biogenesis actually controls cell cycle progression.
How could ribosome biogenesis affect the cell cycle control? It is known that the rate of ribosome biogenesis has been demonstrated to modulate the expression of p53. This occurs through the interaction between ribosomal proteins and MDM2. Some investigations have shown that the large subunit ribosome proteins RPL5, RPL11, and RPL23 bind MDM2 and inhibit the E3 ubiquinate ligase function. In physiological conditions, ribosomal proteins associate with the newly synthesized rRNA to bind ribosomes, and only a portion of RPs is available to bind MDM2, thus keeping the level of p53 low. And the level of p53 rapidly increases with the inhibition of rRNA transcription or maturation, because the ribosomal proteins no longer used for ribosome formation are left free to bind to MDM2, thus blocking p53 protease digestion.
Hi, I'm going to talk about ribosome biogenesis rate and tumorigenesis. So, recent studies indicate that quantitative changes in ribosome biogenesis may be responsible for neoplastic transformation. The stimulation of ribosomal biogenesis by interleukin-6 induces the epithelial mesenchymal transition, EMT, and increases the invasiveness in a human colon mucosal epithelial cell line. With regard to human pathological conditions, many diseases in which ribosome biogenesis is stimulated by locally or systemically released factors, as in the case of inflammatory and metabolic diseases, have been shown to be associated with an increased risk of tumorigenesis. Diseases like diabetes, infection, injury, etc. Hence, chronic inflammation increases levels of interleukin-6 and insulin. Both will increase DNA transcription. In this case, infection by HBV and HCV viruses will produce chronic hepatitis, producing or stimulating the activity of PO1 and 3. So there will be an upregulated ribosome biogenesis and increased risk of cancer onset. Therefore, if we have increased ribosome biogenesis, there will be a downregulating p53 transcription and activity. This picture represents the relationship between ribosome biogenesis and p53. If you see the left arrow with a cross, ribosome biogenesis is decreased and p53 is increased in activity. So there will be a cell cycle arrest and or apoptosis. In the central way, there is a balance between ribosome biogenesis and the activity of p53. So, there is a normal cell proliferation rate without EMP changes. In the last way, or in the right arrow, ribosome biogenesis is increased and the activity of p53 is reduced. So, there is an increased cell proliferation rate with EMP changes.
In this image, we could see two histological sections from two breast cancer samples, and in the image A, it's a sample that was characterized by wild-type p53 and hypophosphorylated BRB, while sample B was characterized by mutated p53 and the later active. Here we could compare the small size of the nuclei in breast sample cancer A with the very large nuclei in breast cancer sample B. In the next image, we could see two sections from human colon histological samples. The letter A is a control sample that is from a healthy patient, and the letter B is an ulcerative colitis sample. Here it can be seen that the nuclear size of the epithelial cells lining the mucosal surface of the control sample is smaller than that of the epithelial cell nuclei of the ulcerative colitis sample. And Finally, in this image, we could see a simplified representation of the relationship between the level of ribosome biogenesis and the cell proliferation rate and neoplastic transformation. In the central row, it's represented the normal levels of ribosome biogenesis and p53 in a proliferating cell, leading to a normal cell proliferation rate without epithelial mesenchymal transition changes. In the left arrow, we can see how a downregulation of ribosome biogenesis leads to an increase in p53 levels, producing a cell cycle arrest and/or apoptosis of the cells, while an upregulation of ribosome biogenesis, represented by the right arrow, have the opposite effect, decreasing the levels of p53 and increasing cell proliferation rate with epithelial mesenchymal transition changes.